When you really boil it down, a virus has just one single-minded goal: make more of itself. But as an obligate intracellular parasite, it's pretty helpless on its own. It absolutely needs our cells. So today, we're going to break down this whole process. Think of it like an intricate, multi-stage biological heist.
So take a look at this electron micrograph. It really shows you just how minimalist these things are. I mean, they're kind of beautiful in their simplicity, right? Just a little piece of genetic code stuffed into a protein coat and maybe wrapped in a stolen bit of membrane. So the big question for us, the one we're going to tackle, is how does this ridiculously simple package pull off such a masterful takeover of our incredibly complex cells?
Okay, so to really get our heads around this, let's stick with that idea of a mission. For the virus, this isn't just a choice, it's a biological imperative. It has one single, non-negotiable objective: replicate. And it'll do that at any cost to the poor host cell.
Right, so here are our and the virus's mission objectives for today. First, we're going to map out the whole sequence of events step by step. Then we'll dive into the real molecular nitty-gritty of how it copies its blueprints. And finally, we'll really focus on the two most important parts of the whole operation: making new genomes and making the proteins to package them up.
All right, so every good heist needs reconnaissance, right? You've got to case the joint. And it's no different for a virus. Before it can even think about replicating, it's got to get into the host, and then, and this is crucial, it has to find the exact right cell to infect. This is that casing the joint phase.
So on the big picture organismal level, the virus needs a door. The most common ones are, of course, mucosal surfaces. You've got your respiratory tract, you've got your GI tract. Think of influenza spreading through aerosols, or enteric viruses using the good old fecal-oral route. And of course, any break in the skin, an abrasion, an insect bite, a needle, that's another open invitation.
But just getting inside the body? That's not enough. The virus has to find its specific cellular target. And this is where a really key concept comes in, called tropism. Think of it as a super-specific lock-and-key system. It's what dictates which cells a virus can or can't infect, and it all comes down to this molecular handshake between proteins on the virus surface and receptors on our cells.
And this table here, it lays it out perfectly. Look at HIV-1. It's going specifically for CD4 positive T cells, and it does that by binding to the CD4 receptor and some chemokine co-receptors. Influenza A, on the other hand, it's looking for sialic acid on your respiratory cells. Or rhinovirus, which causes the common cold, it uses ICAM1 on your nasal epithelium. What's the common theme here? In every single case, the virus is cleverly exploiting proteins that are already there, doing a normal job for the cell. And these attachment strategies can be pretty different in their complexity. So for something like influenza A, it's pretty straightforward. It just needs to find that one receptor, sialic acid, and it can latch on. But then you look at HIV-1, and it's much more sophisticated. It's a two-step process. First, it binds to its primary receptor, CD4. That binding actually causes a shape change, which then lets it grab onto a second molecule, a co-receptor, like CCR5. It's like needing two keys to open a lock, which gives it this extra layer of specificity.
Okay, so the virus has found its target and it's latched on. Now for the next big challenge, getting its payload, that precious genome, across the cell's plasma membrane. This is the infiltration phase of our heist.
Right, so there are two main ways it can do this. The first is direct membrane fusion. This is a tactic used by enveloped viruses, and it's pretty much what it sounds like. The viral envelope just merges with the host's membrane. It's a really clean, direct way in. And what's really important here is that it's pH independent. That means it can happen right there on the cell surface where the pH is neutral.
And we can see that exact process here with HIV. So after it does that two-step receptor binding we just talked about, the viral and cellular membranes fuse together. And bam, the viral core gets deposited right into the cytoplasm. You can see how the viral envelope proteins just get left behind, almost like a calling card stuck on the cell surface.
Now, the second main strategy is a bit more sneaky. Here, the virus actually tricks the cell into swallowing it whole. It co-opts a normal cellular process called endocytosis. So the cell thinks it's just taking something in from the outside, but it's actually engulfing the virus particle, trapping it inside a little bubble called an endosome. But the virus isn't home free just yet. Now it's stuck inside this membrane-bound prison.
And this is where it gets really clever. Let's take influenza as our example. As that endosome moves into the cell and matures, the cell naturally starts pumping protons into it, which makes the pH drop. And that drop in pH, that's the signal. It's the trigger the virus was waiting for. It causes a massive shape change in the viral fusion proteins, and they spring into action, fusing the viral envelope with the endosome's membrane. And just like that, the viral genome is released into the cytoplasm. It's just a beautiful example of a virus exploiting a totally fundamental cellular pathway for its own ends.
So, let's recap. The virus is in. Its genome is free in the cytoplasm. Now, the main event begins. It's time to execute the plan. And this part is all about one thing: a complete hostile takeover of the cell's resources to churn out thousands of copies of the viral genome and all the proteins it needs.
Okay, so when it comes to replication strategies, there's a whole bunch of them. And the way we make sense of it all is by using the Baltimore classification system. Now, the single most important thing to remember here is that all roads lead to messenger RNA. I don't care if the virus starts with double-stranded DNA, single-stranded RNA, whatever, it has to make mRNA. Why? Because mRNA is the only language the host cell's protein factories, the ribosomes, can understand. So this whole classification system is just based on the different pathways viruses take to get to that crucial mRNA.
So, let's dive into this with a case study. First up, poliovirus. It's a class 4 virus, and its genome is a single strand of what we call positive sense RNA. Now, that term positive sense is absolutely key. It means the viral RNA is, for all intents and purposes, already an mRNA molecule. It can be read directly by the host ribosomes, no extra steps needed.
Let's follow along on this diagram here and trace what happens. So look at step 3. The genome gets into the cytoplasm and boom, it's immediately grabbed by the cell's ribosomes. They read it and translate it into one giant protein. We call it a polyprotein. This massive thing then gets chopped up into individual functional proteins by viral proteases. One of the most important ones is the RNA-dependent RNA polymerase, or RDRP. This new enzyme then goes and hijacks bits of the endoplasmic reticulum, creating these little viral factories. Inside these factories, it makes negative sense copies of the genome, which then act as templates to just mass-produce thousands upon thousands of new positive sense genomes. It's an assembly line.
So just to quickly summarize the tactical playbook for poliovirus, first, its genome is infectious all by itself. It uses a little protein called VPG as a primer to get replication started. It brutally shuts down the host's own protein synthesis so it can have all the ribosomes for itself. It builds those dedicated viral factories on hijacked membranes, and it uses that double-stranded RNA intermediate to just crank out new genomes. I mean, it is an incredibly efficient takeover.
Okay, so that naturally leads to a question, doesn't it? What happens if a virus has an RNA genome, but it's the wrong sense? What if it's a negative sense strand? To a ribosome, that's just molecular gibberish. It can't read it. So what does the virus do then?
Well, that brings us to our second case study, the negative sense single-stranded RNA viruses. Think rabies, for example. Since their genome can't be translated directly, they have to come prepared. They bring their own tools. Tucked right inside the virion, already made, is their own RNA-dependent RNA polymerase. As soon as the virus gets in, that enzyme gets to work, using the negative sense genome as a template to make readable positive sense mRNA strands. Then, the host ribosomes can jump on and translate those into viral proteins, including, of course, a whole lot more of that polymerase enzyme.
This slide really nails the comparison. For polio, a positive sense virus, the genome is the message. For rabies, a negative sense virus, the genome is just the template for the message. So what does that mean? Polio gets translated immediately. Rabies has to be transcribed first, and this leads to the absolute most critical difference, and this is a key takeaway. A negative sense virus must carry that polymerase enzyme with it, inside the virion. If you just injected the naked RNA from a rabies virus into a cell, nothing would happen. It's not infectious. But the naked RNA from poliovirus, that is infectious. That's a huge distinction.
Okay, so at this point, the host cell is just stuffed to the gills with new viral genomes and viral proteins. It's done its job. So the final phase of our heist can begin, the getaway. This is all about assembling all those new parts into brand new, infectious virions, and then getting the heck out of the now doomed host cell.
Now, assembly isn't just a random jumble. It's a highly organized production line. First, all the components, the proteins and the genomes, they all get concentrated at a specific spot in the cell. Some viruses, like the herpes viruses, even build a temporary protein scaffold to guide the whole construction process. Next, the viral genome gets carefully packaged into the forming capsid, and then there's a final maturation step.
This often involves viral proteases that come in and make a few final SNPs, which causes the structural proteins to snap into their final, stable, and most importantly, infectious shape.
And that really brings us to our final thought for today. This whole process, it's a constant, high-stakes evolutionary arms race. Our immune system comes up with new defenses, and the viruses, especially these RNA viruses with their sloppy, error-prone polymerases, they evolve incredibly fast to get around them. So, now that we've walked through this whole intricate molecular dance, the question I want to leave you with is this. When you look at all these incredibly specific, essential steps, the receptor binding, the fusion, the replication enzymes, the protease activity, where do you think the best targets for the next generation of antiviral drugs are hiding? Figuring that out? Well, that's the challenge for you.